Hello, could somebody confirm that you can hear me?
I can hear you.
Thank you. Close that down. All right, what we're going to do today is just review for the final. Are there any questions before I begin? Actually, before I begin, let me talk about the gradebook. Am I sharing my screen? No. So let me go to the gradebook. I think I have now all of the assignments graded except for the unknown project and the infectious disease project. Please remember you need to finish your unknown notes and turn in your unknown project. Well, the unknown notes page will be closing on Friday. The unknown project is due this Saturday. Any questions about any of that? All right, let me go to the grade books so I'm not sharing my screen. I got the last of the study, the extra credit for the study groups done. I got that graded. I also put up the correct score for quiz. What was that, 4B? Something like that. So you now have full points up for quiz 4B. And I have the drop quiz in there as well. I need to finish worksheet lecture chapter 7, but I don't think that needs to be turned in until this Saturday. Nope, but actually it's due today, so I can't get it graded after today. So I have the drop quiz in there, meaning I figured out which was your lowest quiz and put it in the drop quiz column. You'll note that I gave you 6 points of extra credit, and instead of dropping your lowest quiz score, I dropped your lowest quiz score and then added 6 points of extra credit. Any questions about that? Still not seeing which one it was I got graded here. No, it's not A, it has to be B. I think it would. No, it's quiz 5. All right, quiz 5. I now have quiz 5, which was out of 0.24 points. I simply multiplied that to make quiz 5C, and you add quiz 5 and quiz 5C columns together, it'll be a total of 75 points. Is that right? Come down to, yeah, it's going to be 75 points. Any questions about any of that? So all your quizzes are now showing and graded, and I have the drop quiz, and your lowest quiz score will be subtracted in the drop quiz column. With the exception, you're getting 6 extra credit points in the drop quiz column. So now the total quiz points is correct. You are counting the quiz. Let's say quiz 1 was dropped, quiz 1A and quiz 1B is dropped. And so that'll mean in the drop quiz score. And so it's added in quiz 1A and quiz 1B and subtracted in the drop quiz column. Any questions about any of that? So I looked at the scores, and actually you guys are doing very well. Over 50% of the class have A's so far going into the final. Any questions about anything? So good job. If you have any comments or questions or concerns, please contact me. I am here to help you. All right. If there's no questions, let me get out of the grade book and then I'll share my screen. Scroll up here. I'm not seeing how to get, I'm not on the top yet. There we go. So I'll try and get the infectious disease project graded this weekend. And then you'll know its grade before you take the final. Let me briefly talk about the final. There will be two parts. Final A, which will be a fill in the blank or short answer question. There's only a few questions in this. It's not timed, but it must be completed by 11:59 p.m. of the day of the quiz. And you can take it starting June 13th at 11:59 p.m. to June 14th at 11:59 p.m. So the final A is not graded, it's short answer and fill in the blank questions. There are only 1, 2, 3, 4, 5 questions in this one. This includes questions on your unknown report. So it's got information about like identifying the bacteria. All right, that's final A. Final B requires Respondus Lockdown Browser. It is a time test. It's worth 135 points. The time limit is 70 minutes. That's 1 minute per question, plus, I don't know, some extra minutes. I don't know exactly how many, but let me go into there and then I can answer that. Numbered here, so I'm having trouble finding how many questions there are. Well, 135 points divided by 3. What is that? Let me get that up. About 45 questions. I'm close. Okay, any question about the finals? All right, if not, let's begin the review. Close that down. Let me share my screen. I'm going to start at the bottom, just because there's going to be more questions. I can mention that about the final. Let me go up and call this up. The final 50% of the questions will come from the material that you have not been quizzed on already. So of the final, there will be heavy coverage of chapter 7, 13, 14, and lab module 9. Also on the final is the unknown project. There will be a question or two about the unknown project. And then the other 50% of the final will be over the rest of the material in the course, starting from chapter 1 and going through chapter 8, which you have been quizzed on before. And that includes lab 00, to lab 8. So let's begin with chapter 7. So you should be able to discuss the factors that influence microbial death rates. You should be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells. You should know a little bit about the various types of disinfectants and how they work, and understand the relative resistance of the major microbial groups to antimicrobial agents. When we're talking about the antimicrobial groups and the relative resistance, you should know the different characteristics of some of them, like endospores can survive many different agents, antimicrobial agents, because of their tough endospore coating. But there are a few agents which do kill endospores. And the one that comes to mind most readily is hydrogen peroxide and the other peroxides, because endospores are very sensitive to oxygen, meaning all of the endospore forming organisms are anaerobic or facultative anaerobes. The mycobacteria, another bacteria which is highly resistant to most of the antimicrobial agents, and that's because of the mycolic acid in the cells, not allowing the agent to come into the mycobacterium and then do damage to the cell. But there is one agent which is actually a lousy disinfectant, which is very good at killing mycobacteria, and that is the alcohols like ethanol. And that's because it goes right through the mycolic acid. Any questions about microbial characteristics and the microbial control? All right, let's see if there's anything else I want to mention. Read this. You should know that phenol is, of course, one of the phenols and phenolics, and that Joseph Lister used that as the basis for his phenol coefficient, meaning everything that's a disinfectant it has a certain phenol coefficient. And in his day, phenol was the highest chemical he found that had killing effects on microbes. And he used phenol to treat his surgical patients. So he used it to make the surgery aseptic. And phenol is actually a lousy chemical to use because first, it's not that good at killing cells. The phenol coefficient is only 1. And when we talk about the modern agents we use in the hospital, like for surgery, the phenol coefficient is something like 1,000. So 1,000 times better than phenol. And then the other reason why phenol would be a bad substance to use on surgical patients is phenol is irritating to the skin. It's also corrosive on surgical instruments, metal instruments. And then phenol is a carcinogen. So if you apply phenol to the patient's skin before surgery, you may have a successful surgery, but then later on the patient may come down with cancer. Some halogens you should know, there's 2, and that's tincture of iodine or iodine, and then bleach. Those are the most commonly used halogens. We already talked a little bit about alcohol. I think those are the only things I want to mention from this table. Hydrogen peroxide is a peroxygen, and I mentioned that it kills by oxidation, and it's very good at killing endospores. So that's something else you should know from here. You should know that there's two forms of radiation which can kill cells. Ionizing radiation, which gives molecules and atoms high energy, causing the electrons, once they absorb that high energy, to leave the atom, causing ions. The ions can then go around and damage the cell, such as damaging DNA. And then there's non-ionizing radiation, of which UV light is the one that does damage to DNA. Another non-ionizing radiation would be visible light. It doesn't damage DNA, but UV light does. It causes thymine dimers in two thymines that are next to each other, and that damages the DNA. This part of the DNA cannot be replicated or transcribed because it has a thymine dimer right there. You should know that heat is something we can use to control microbes. And there's two types of heat, dry heat and wet heat. This picture is trying to show you that dry heat, the cat climbing into the oven, is not as efficient at transferring the heat and then killing the microbes as wet heat is. Any question about that? All right, you should know a biocide is a substance that kills microbes, such as a bactericide, which kills bacteria. Bacteriostasis is an agent that inhibits the growth of the microbe. And once you remove the bacteriostatic agent, the microbe may continue to grow. If it's still alive, it probably would continue to grow. There are some bacteriostatic antibiotics, and how they work is they prevent the microbe from growing. And then our immune system removes the microbes from the body while the bacteriostatic agent is acting. Any question about that? You should know that there are different things that can affect the microbial death rate. The number of microbes, the more microbes you have, the longer the exposure to the antimicrobial agent must be to get the same killing. Environmental influences can affect the microbial death rate. Mainly organic matter can inactivate the antimicrobial agent. This is especially true with bleach. It is highly inactivated by organic matter. And you should realize that in a hospital setting, organic matter is oftentimes around, almost always around. The time of exposure affects the microbial death rate, and then the microbial characteristics. For example, we do have killing of endospores with an agent. However, the time required to kill the endospores will be much greater than if the cells did not have endospores. Any questions about that? All right, I think that's it for chapter 7. Let's move on to chapter 14. In this chapter, you should really know the terms of this chapter, because if you know the terms of chapter 14, you're going to learn half of the chapter. Most of the material in this chapter is just terms. You should also understand the relationship of the normal microbiota and its host. Everyone needs a normal microbiota to maintain their health. Our normal microbiota helps us digest food. gives us certain vitamins, and then protects us from many human pathogens. We'll talk about how the normal microbiota can compete with the human pathogen in just a little bit. You should also understand the stages of disease development and what nosocomial infections are and how to control them. In fact, this is one of the best things you can learn if you're going into the healthcare industry, because nosocomial infections are something a patient gets when they come to a healthcare facility. They never had them until they came to the healthcare facility, and then they're picked up, and they're important because a lot of deaths in the United States, as well as many patients, about 10% of patients in the United States get a nosocomial infection. So we really need to do better at preventing them. And I have to say that in the last 10 years, the number of nosocomial infections in the United States has been reduced by about 50%. So we have done better. And the number of nosocomial infections from sometime long ago, like the 1960s, has really improved. In the 1960s, the number one nosocomial infective agent was Staphylococcus aureus. And who was spreading it? The healthcare workers. They'd be touching one patient, not washing their hands, not washing their stethoscope like a doctor, and then spreading the Staphylococcus aureus from one patient to another. And it used to be the number one nosocomial infective agent. Now it is no longer, because of steps we've taken, especially in the healthcare people. They're washing their hands after touching a patient, they're wearing gloves, etc. And so Staphylococcus aureus is no longer the number one nosocomial infective agent. The number one nosocomial infective agent today is, of course, E. coli. And as you know, we all have E. coli. And so it's very difficult to totally control that one. But we need to prevent the spread of E. coli, and then having the E. coli causing a nosocomial infective agent. So you need to really learn that just because of your career choice. Any questions about any of these? All right, you should know the location of the normal microbiota in the human body and how the normal microbiota is different from a transient microbiota. A transient microbiota is an organism that a person has for a relatively short duration. It may be days, weeks, or months, but unlike the normal microbiota, it does not permanently or nearly permanently colonize the host and then remain with the host for most of their lifespan. You should know we have more normal microbiota cells, more bacteria cells in our body and on our body than we have human cells. All right, the normal microbiota can engage in microbial antagonism with a human pathogen, such as preventing the human pathogen from occupying a niche. Like if I were to get a human pathogen to fall on my skin, it probably would not get established because I've got lots of normal microbiota on my skin. The normal microbiota can compete for resources as well as space. The normal microbiota can produce acids, which would make the environment too acidic for the human pathogen to get established. A place which is more acidic than normal would be our skin. The normal microbiota makes our skin more acidic than it would be without the normal microbiota. And then another place would be the urethra, especially the lower urethra, the normal microbiota, make it somewhat acidic, and it oftentimes is too acidic for a human pathogen. And then the normal microbiota may also produce bacteriocins, which are proteins that act like antibiotics, except their spectrum of action is very narrow. Like a bacteriocin produced by E. coli will stop the growth of Salmonella and Shigella, but it would not stop the growth of other bacteria in the gut, such as, is it Enterococcus faecalis? No, used to be Enterococcus faecalis. Used to be known Streptococcus faecalis. You should know the normal microbiota lives in symbiosis with us, forming either mutualism, where both organisms benefit, or commensalism, where one organism benefits, probably the normal microbiota, but we're not harmed. Parasitism is where the microbe benefits and we are harmed, and all human pathogens form a parasitic symbiotic relationship. You should know about Koch's postulates. We've talked about it at least 3 different times in this class, so you can count on a question about Koch's postulate being on the final. I think you should know the difference between a communicable disease and a contagious disease, and then give examples. All right, you should know the stages of the development of disease. When a patient first becomes infected with a disease-causing organism, and before the patient has any signs or symptoms, we call that the incubation period. This is where the organism is growing in the patient, but hasn't reached the threshold to cause signs or symptoms. Then we have the prodromal period, where the patient now has mild signs and symptoms. After that, we have the period of illness, where the signs and symptoms are most severe. And this is also where the disease-causing organism has the highest number in the patient. That's followed by the period of decline, where the patient starts feeling better, but they still have signs and symptoms. Once they no longer have signs or symptoms, that begins the period of convalescence. And that period lasts as long as the disease-causing organism is in the patient. It doesn't end until the disease-causing organism is no longer in the patient. So the period of convalescence is at the end. At all the periods, you can spread disease. What else did I want to say? Oh, and that is some diseases lack certain of the periods. Like when you get a flu, you may never have a prodromal period. And then some diseases, especially those that kill patients, they never go past the period of illness, meaning the patient dies, and they never enter a period of decline or a period of convalescence. There are also some diseases that do not kill the patient, like hepatitis B and hepatitis C infection, and the patient always has the disease, and so they're always in the period of illness. Oh, come on. You should know about transmission of disease. There's contact transmission, vehicle transmission, and vector transmission. I already mentioned you should know about nosocomial infections and how to control them. All right, any questions about chapter 14? Not, let's move on to chapter 13. You should know what viruses are, what viroids are, and prions. You should know the different terms to describe them. You should know, in particular, the different life cycle that viruses have. Bacteriophages have the lytic life cycle and the lysogenic life cycle. Animal viruses have the lytic life cycle. And the only one I'll quiz you on, well, I take that back. There's two of them you need to know. There's the budding life cycle, where the virus buds out of the cell or the cell membrane or the nuclear membrane or the endoplasmic reticulum, the membrane of the endoplasmic reticulum. The budding life cycle virus usually, almost always, does not kill the cell. Usually, if the cell is killed, it's the host immune system that recognizes that cell is infected and then kills the cell. The third animal virus life cycle you should know is the latent life cycle. In the latent life cycle, the virus goes dormant, just sits out, doesn't reproduce while it remains in latency. It doesn't reproduce, and it's sort of in stasis, sort of like hibernation. It's not doing anything. It can come out of latency and then enter the budding life cycle, in which case it causes disease. So you should know those three life cycles. You should know about the characteristics and structure and classification of viruses. The virus goal is to replicate, and they need a host cell to replicate. They cannot do any replication outside of the host cell. In fact, they have little or no metabolic activity outside their host cell. They require their host cell to multiply, so we can call them an obligate intracellular parasite. You should know the host range is the range of cells that can be infected with a virus. Most viruses only infect certain cells of about one species. Like HIV can infect T cells. I think they can infect brain cells and macrophages or monocytes. So they can infect those cells, and they don't infect other cells like skin cells or bone cells. How that is determined is by the molecules on the surface of the virus that we call ligands or adherins, which bind to the molecules of the host cell, and we call these molecules receptors. So the virus will bind to the receptor. Any question about any of that? You should know that viruses have a nucleic acid for their genome. It can be either DNA or RNA, but not both. The DNA can be double-stranded DNA, single-stranded DNA, or it can be RNA, in which case it can be double-stranded RNA or single-stranded RNA. If ever we're talking about single-stranded RNA, it's important to state whether that strand, single strand, is positive strand or the negative strand RNA. The positive single-strand RNA may be read by the ribosome as a messenger RNA. The negative single-strand RNA cannot be read by the ribosome. You should know the taxonomy of viruses. I mentioned the life cycles. One consequence of lysogeny, let's go on, is that a lysogenic cell, meaning a bacteria cell which has a lysogenic virus in it, is going to have new DNA that the cell originally did not have. And that can give the cell new properties. The best example is Corynebacterium diphtherium only produces the diphtherium toxin when the bacteria has a lysogenic virus within it, because the lysogenic virus codes for the Corynebacterium diphtherium toxin. You should know that animal viruses are similar to bacteria viruses or bacteriophages in their infection and replication. There are some differences, the main difference being an animal virus can multiply in the cytoplasm or it can multiply in the nucleus. Bacteriophages can only replicate in the cytoplasm. You should know that retroviruses are an exception. This is an RNA virus that does replicate in the nucleus. And that's because it has a DNA intermediate, meaning the virus is RNA, positive single-strand RNA. And when the virus replicates, it has a DNA intermediate, which then integrates into the host cell's chromosome. And then to replicate, that DNA is transcribed to make the RNA, it's positive single-strand RNA. That positive single-strand RNA is the genome of the virus. It also can be used as messenger RNA and translated into the viral proteins, which then get together to make the new virion, which can then bud out of a membrane in the cell. So retroviruses are unique because they have the enzyme reverse transcriptase that can copy or make DNA from RNA. You should know a little bit about coronaviruses, mainly that SARS, MERS, and COVID-19 is a coronavirus. You should also know that the second most common cause of the human cold are coronaviruses. They're not the number one cause of human colds, but they're the number two cause of human colds. So there are coronaviruses that give people colds. And actually, coronaviruses tend to give children colds, meaning usually you get the coronavirus when you're very young or a child. You should know that the transmission of COVID-19 is primarily via droplets from coughs or sneezes within about 6 feet. However, the coronavirus can spread in other ways. The virus can go beyond 6 feet. You could get it simply from talking to someone. You can also pick it up on a surface. Fortunately, COVID-19 does not tend to spread from surface to surface, meaning you touch like a doorknob with it and put the COVID-19 on the doorknob, then the next person who touches the doorknob picks it up on their hands, and then they inoculate a mucous membrane in their face with their hands, and that's how they get infected. And that's the way many colds and flus actually spread, from indirect contact, meaning from a fomite. COVID-19 can spread that way. Fortunately, it prefers to spread patient to patient. So most COVID-19 is picked up patient to patient. You should know an acute viral infection will happen when the virus quickly infects you and then quickly reproduces, increasing in number, and then quickly leaves. The best example is the human cold, where you may be infected by one or just a few viruses. They grow within you, quickly multiplying, and then quickly leave. That's very different from a persistent viral infection. When the patient first gets it, they have a very low load of the virus, and they have very mild signs and symptoms, and the virus over time just builds up. So the signs and symptoms increase in number and severity as the viral load increases. Oftentimes in the persistent infection, we'll have a bloom where there'll be a lot of virus coming out, and then the patient will die, so the curve ends. That isn't true for all persistent viral infections. It used to be true for the way AIDS developed, but nowadays we have modern medication which can make an AIDS patient live almost a normal lifespan, meaning it can make an AIDS patient survive into old age, and they may die a few years earlier than a non-AIDS patient, but the point is they have close to a normal lifespan. And then let's talk about an acute infection would be something like when the child gets chickenpox, and then the virus is latent, so you don't see any in the patient. It's hiding out in their ganglia nerves, at least the chickenpox virus, but it's not replicating, not causing any signs or symptoms. But then it can come out, in which case we call it a latent infection. And in the case of chickenpox, the very first time you get it is chickenpox. All other times you get the disease where the virus is replicating, we call it the shingles. All right, you should know about prions, how prions replicate, and then you should know about viroids, which are plant RNA molecules that are infectious. Any question about any of that? All right, let's move on. Close that down. Chapter 8. Chapter 8 is an important topic. As you notice, we spent about 3 days on Chapter 8, and that's because genetics is very important to microbes as well as all organisms. So know the terms of Chapter 8, be able to describe DNA replication, RNA transcription, and protein translation. Also understand the different types of mutation, what they are. So you should know what the genotype is and a phenotype. You should know DNA typically in the cell is double-stranded. One strand is held together by polar covalent bonds between the sugar and the phosphate, holding the strand of DNA together. The two strands are held together only by the hydrogen bonds between the nitrogenous bases, meaning the nucleotide bases of DNA. And this bond is very weak, but we have many hydrogen bonds holding two strands of DNA together. And that's why in body temperature, even though the DNA hydrogen bond, this one can break and then reform all the time, it's held together by the other hydrogen bonds. So DNA is normally double-stranded in our cells. There is an exception, and that is when the DNA is being replicated, it is single-stranded, or when the DNA is being transcribed, it is also single-stranded. You should know the central dogma. You should know the flow of genetic information in the cell. And within a cell, between generations of cells, and you could say you should know the flow of genetic information between cells of the same generation. I don't think I have any questions about RNA processing, but in eukaryotes, the RNA has to be processed before it can leave the nucleus and then be, like if it's messenger RNA, before it can be translated. So before it can leave the nucleus, the introns have to be spliced out of that original RNA transcript, and then the exons have to be spliced together before we get our finished RNA product. You should know about what a bacterial operon is and how they can control gene expression. You only need to know about the lac operon, which is an inducible operon, where there is a different gene, the I gene, which makes a regulatory protein, and it can bind to the O of the operator to shut down the expression of the structural genes Z, Y, and A, Z being the lactase enzyme. P, of course, is part of the operon, and it is where the RNA polymerase binds to the DNA, and then will transcribe the RNA after it binds to the P. How the lac operon is regulated is the I gene makes a repressor protein which binds to the operator, and then that prevents the RNA polymerase from moving further down than the operator, so it's stopped right here. And then the Z, the Y, and the A gene are not transcribed. Normally, in E. coli, the Z, the Y, and the A gene are not transcribed. They are only transcribed when the inducer, allolactose, which is just an isomer of the sugar lactose, is around. And allolactose can bind to the repressor protein and inactivate it. And if there's no repressor protein, the operator is now free. And so the Z, the Y, and the A gene can be transcribed. Any question about any of that? All right. One thing you should remember about mutations. See if I can get back here. Oh, come on. There we go. Here it is. Some of the types of mutation happen because of the table of the codons. Where's my slideshow? There we go. Meaning a silent mutation can happen whenever the codon UUU is changed to UUC. We call it a silent mutation because there is a change in the DNA and a change in the RNA, but it does not change the amino acid of the protein. So that mutation is called the silent mutation. If ever you change the DNA, which results in a change in the amino acid, such as changing UUU to UCU. This is a change in the DNA and a change in the amino acid. So this is a missense mutation. Any question about any of that? Let's end that. So that's a missense mutation. If the DNA is mutated and then the reading frame of the protein, the codon, is a stop codon, that we call a nonsense mutation. You should know what a frame shift mutation is. And then once you have a frame shift mutation, the codon is changed in that protein from the point of the frame shift mutation. Downstream will be totally changed if you add one or two nucleotides, or you subtract one or two nucleotides. If you subtract or add 3 nucleotides, you don't have a frame shift mutation. You do have a mutation because, like if you're subtracting 3, there's no amino acid there in the position in the protein. But it is not a frame shift mutation because the rest of the protein has the same frame as the original protein. You should also know what a macromutation is and pay attention to my lecture because your book doesn't discuss it. It's where an entire region of a chromosome or an entire chromosome is either added or deleted. Oops, wrong way. You should know the causes of mutations. There's 2 types of mutations, ionizing and non-ionizing radiation. We already discussed that, so I'm not going to discuss it further. You should know what recombination is and how it's slightly different in Meyer general than crossing over. And then you should know that once a gene exists in a population, that gene can spread around by vertical gene transfer. That gene can spread around by horizontal gene transfer, including by transformation, conjugation, and transduction. In transformation, a part of a chromosome comes out of a cell, is naked DNA in the environment. That naked DNA is taken up by the recombinant cell, and then a portion or all of that DNA is incorporated into the chromosome, and then this recombinant cell has a new gene, the lowercase a, which came from this strand of DNA, which came from another cell. The second way horizontal gene transfer can happen is by sexual conjugation. In gram-negative cells, we have a sex pilus, which skewers a gram-positive cell, brings the 2 cells closer together. They do not need to be like this close because the sex pilus is hollow, and then the plasmid from this cell can replicate and then move through the sex pilus to this cell. That will change this phenotype from F- to F+. And this cell will then grow a sex pilus. The third way that horizontal gene transfer can happen is using a virus. Now, this will be a little clearer to you because we've discussed virology. What happens is that when viruses multiply, mistakes get made. Viruses aren't worried about it because they have lots of offspring. And if one of them isn't going to work, the cell just does not worry, or the virus doesn't worry about it. The thing is that this virus, instead of getting viral DNA stuffed in it, it got DNA from this chromosome, meaning the DNA was degraded, but it's in chunks, and a chunk got input into this virus, protein code, I guess I should say, not a virus. And then when this virus infects this cell, it injects its genome into this cell. This cell is not getting viral DNA. This virus is not going to replicate in this cell. What's going to happen is this DNA can integrate into the bacterial chromosome and then bring new genes to this cell, the recipient cell. And those genes came from this cell. All right, those are the three forms of horizontal gene transfer. I was wondering if I want to talk about one other thing here, but I think not. All right, you should know that bacteria cells can have a small molecule of DNA, which is not a part of the chromosome. We call it extrachromosomal DNA, and we call that a plasmid, the extrachromosomal DNA. Now, in human cells, cells do have DNA outside of the nucleus, outside of the nucleus. We don't usually call it extrachromosomal DNA, where this DNA is in the mitochondria and in the chloroplast. And usually we call those mitochondrial or chloroplastic DNA. It's actually the genome of the cell that a long, long time ago started living in symbiosis with the proto-eukaryotic cell. And what I'm referring to is the theory for how the theory for how chloroplasts and mitochondria came about and started being a part of the eukaryotic cell. You should know about transposons, what they are, and how they can move around from one position in the DNA to another position. All right, any questions about chapter 8? Not, let's go on to chapter 6. You should know the terms of this chapter. You should know the difference between a reproductive spore, where one cell makes more than one reproductive spore, and an endospore, which is not reproduction, because one cell makes one endospore, and then the parent cell dies. So that's 1-to-1, it's not reproduction. And you should know that endospores can survive many harsh environments. Reproductive spores are not, their goal is not to aid survival in a harsh environment. Their goal is simply to reproduce. You should understand the various requirements for growth and understand the 4 phases of growth. All living things have requirements for growing, and there are physical requirements such as the organism must be in the correct temperature, the correct pH, the correct salinity. If they are not, then they may not grow. And if they're in really harsh environments, the cell will die. Like the pH is wrong and way off, the cell will die. But if it's in the correct pH, then the cell will live and will grow. There are also chemical sources required for microbial growth. All living things require a chemical source, a nitrogen source, a sulfur source, a phosphorus or phosphate source, trace elements, and then some oxygen requirements, either with oxygen, with reduced oxygen, or no oxygen, and other organic growth factors. Like for humans, we require vitamin C as an organic growth factor. Microbes, the ones we've been using, don't usually have organic growth factors, but Streptococcus pyogenes is one, and we have discussed it. It cannot grow on a general purpose media because it needs additives that are supplied by red blood cells before Streptococcus pyogenes will grow. Any question about any of that? All right, you know the different temperature that organisms can grow at. You know what an acidophile is. You know what a halophile is. You should realize that all living things have a carbon requirement. Half of all living cells are, when it's dry weight, half of all living cells are carbon. There are two sources of carbon. You can take carbon dioxide either from the air or the water, in which case you're an autotroph. And then you can get carbon from an organic chemical molecule, such as we get our carbon needs from eating glucose and related food molecules. All right, I talked about these. You should know the 4 toxic forms of oxygen. You should know the 5 types of microbial growth with respect to oxygen. Let me blow this up. Come on. If the organism primarily grows at the top of the thioglycolate tube and the reduced growth as you go down the tube, this is an obligate aerobe. If, on the other hand, the organism primarily grows in the bottom of the tube, and as you go up the tube, there's less growth, this is an obligate anaerobe. A facultative anaerobe has most of the growth at the top, and as you go down the tube, there's less and less growth, but you do get growth everywhere in the tube. The reason why there's more growth at the top of the tube is that facultative anaerobes can engage in aerobic respiration. And because anaerobes are organisms that can engage in aerobic respiration, make the most ATP, these cells grow faster in this region of the tube than any other region of the tube. There's also aerotolerant anaerobes, and these are microbes that have all the enzymes they need to detoxify the 4 toxic metabolites of oxygen. So they can detoxify oxygen, but they do not need oxygen for their growth. And so these organisms grow everywhere in the tube. There's also the microaerophiles. They are aerobes, so they do require oxygen to grow, but they will grow in the middle of the tube because they do not like 21% oxygen, which is in the Earth's air. And we'll see most of their growth in the middle of the tube. So they do require some oxygen to grow, but they don't want full oxygen to grow. An example of a bacteria that is a microaerophile is Helicobacter pylori, which is in our stomachs, and partly because the sphincter is closed and we have microbes in our stomach which use oxygen, that reduces the oxygen in the stomach. And then the Helicobacter pylori can grow. All right, any question about any of that? You should know what an auger is and the different types of augers. You should know the purpose for streaking a colony to get isolated colonies. meaning colonies that are not touching other colonies. To get a pure culture, you need to send your cells through 2 colony formation events. And then you'll have a pure culture greater than 95% of the time. If you only streak out 1 time, you will only get a pure culture about 70% of the time. Most bacteria grow by binary fission, but a few do grow by budding and a few by producing reproductive spores. All right, any question about chapter 6? Let's move on to chapter 5. You should know, I don't have a introductory slide for this one. That's odd. I must have made it after I set up spring term. Boy, that's not believable. I'll have to make that a note and make an introductory slide there. Obviously know the terms for this chapter. No idea why I'm just catching this now. Oh, this isn't the right file, that's why. Let me get the other one. There we go, there's my introductory slide. You should know what feedback inhibition is, and in this chapter you should know aerobic respiration. There probably will be a question about aerobic respiration on the exam. So you should know metabolism can be split into catabolism, anabolism, and know what catabolism is and what anabolism is. I guess we should state that all chemical reactions occur by the collision theory, that when atoms, ions, and molecules move about and collide with each other, they can cause a chemical reaction. They will cause a chemical reaction if the atoms or molecules collide with enough energy such that they reach the activation energy required to convert the products, the substrates into the product. An enzyme speed up a chemical reaction. You should know the trophs, that all living things can be classified by their energy source and their carbon source. When we put them all together, we have photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. We are a chemoheterotroph. We get our energy needs from a chemical, and we get our carbon source from an organic chemical. Usually the same, like glucose can be for us both an energy source and a carbon source. That isn't always the case. There are some microbes, prokaryotic cells, that can use an inorganic chemical to get their energy needs. And then they use an organic compound like glucose for their carbon source. These are mostly the chemosynthetic bacteria. However, I should warn you that there are some chemosynthetic bacteria that are chemoautotrophs, and they get their chemical energy needs from an inorganic chemical, and they get their carbon from CO2. Another easy one is the photoautotrophs. They get their energy needs from light, and they get their carbon source from CO2. The photoheterotrophs are ones you're not familiar with. They get their energy needs from light, and they get their carbon source from an organic chemical compound such as glucose. Now, we separated things into these four groupings, but you should realize that life is complex and life doesn't always follow, I don't know, the rules. For example, euglena, when you have it in light and in water and you have no food around for the euglena to eat, it will act as a photoautotroph, getting its energy need from light and its carbon source from CO2. However, if you put glucose into the water and you shine light in, euglena can act as a photoheterotroph, getting its energy need from light and getting its carbon from the glucose. If you now remove the light and have the glucose in there, then euglena will act as a chemoheterotroph, getting its energy needs from glucose and its carbon source from glucose. And my point is that euglena can act as a different troph depending on the environment. Any question about any of that? All right, you should know aerobic respiration. I'm not going to go through it now. Well, I'll go through this. You should know that the summary equation for aerobic respiration, which is many physical and chemical reactions, is C6H12O6 plus 6O2 plus 38 ADP and 38 phosphates derive 6 carbon dioxide, 6 water molecules, and 38 ATP. All the carbon in glucose is converted into CO2. All of the oxygen is converted into water. Any question about any of that? None of the oxygen is converted into CO2. Anyways, we can generate 38 ATP in aerobic respiration. To know the steps of aerobic respiration, there are 3. The 3 steps of aerobic respiration are glycolysis, the preparatory step, and the Krebs cycle, and then the electron transport chain. In a eukaryote, aerobic respiration starts in the cytoplasm with glycolysis and then occurs in the mitochondria. The electron transport chain happens in the inner mitochondrial membrane. In prokaryotes, glycolysis happens in the cytoplasm, so does the Krebs cycle, and the electron transport chain happens in the cell membrane. You should know the different parts of the aerobic respiration, at least the summary equation. You should know about anaerobic respiration and how it differs from aerobic respiration. You should know about fermentation, taking glucose and making ATP, but instead of getting 36 to 38 molecules of ATP in aerobic respiration from glucose, in fermentation, you only generate a net of 2 ATP. And all of those ATP that are generated in fermentation occur from glycolysis, which is the first part of fermentation. All right, you should know in photosynthesis that there's 2 photosynthetic reactions. The non-cyclic photosynthesis, which makes if it's an aerobic non-cyclic photosynthesis, it makes glucose and oxygen. And then there's cyclic photosynthesis that makes ATP. Whatever the cell needs, it will use that photosynthetic reaction, meaning if the cell needs ATP, it'll engage in cyclic photosynthesis. If the cell needs glucose, it'll engage in non-cyclic photosynthesis. Any question about any of that? All right. I'm going to run out of time here, but I'm going to be close. Chapter 4, know the terms. You should know the anatomy of prokaryotic cells. Especially, you should know how gram-positive and gram-negative cell walls differ, and describe the importance of taxis. And then one other thing in this chapter, which we've already talked about briefly, the endosymbiotic theory. Where is that? There we go. The endosymbiotic theory, the theory to explain the evolution of mitochondria and chloroplasts in eukaryotic cells. So you should know the endosymbiotic theory. That's it for chapter 4. Let's move on to chapter 3. Obviously, you should know the terms. You should know the sizes and properties of microscopes. You should know the difference between a light microscope and an electron microscope. And you should know how staining improves resolution. All it does is increase the contrast between the specimen and the background of the slide. You should know that there's 3 different types of stains: a simple stain, a differential stain, and a special stain. Special stains stain specific parts of a cell or specific cell components. The differential stain allows differences to show between cells. And then a simple stain, all it is doing is making the cell more visible under the microscope. You should also understand the gram stain, its procedure, and the basis of its differential staining. Any questions about chapter 3? All right, on chapter 2, I'm going to tell you you don't need to know or study chapter 2 for the final because there's no direct question on chapter 2 in the final. However, I do expect you to understand about the chemistry of biological molecules. So I do expect you to know that, but I'm not going to ask any questions directly from chapter 2. So there are no questions from chapter 2. On chapter 1, you should study this chapter a little bit more heavily than chapters 2 to 8 because chapter 1 introduces many concepts that we later discussed in the term. Like endospores, we first discussed them in chapter 1. The Cox postulates we first discussed in chapter 1. What's the third one? I don't remember. There's something else that we first discussed in chapter 1, and then we discussed it numerous times in the class. And so you should know these topics from chapter 1 and the later chapters, because these are important topics, and that's why we discuss them 3 or more times in the term. Oh, the cell wall. The first time we discussed the cell wall of prokaryotic cells was in chapter 1. The other reason for knowing chapter 1 is it discusses a brief history of microbiology. I think you probably guessed my enthusiasm for the history of microbiology, and I kind of like the history of microbiology. So you can count on a few questions on the history of microbiology. My point is chapter 1 will have more questions than the chapters 2 to 8. simply because it's chapter 1, it is introducing topics, and then it has the history of microbiology. And the history of microbiology is just fascinating. And so it's good to know the history of microbiology. With the history of microbiology, you don't need to know any dates except for when the first fossil evidence for the ancestors of bacteria show up in the fossil record. That date you should know. All the other dates you do not need to know. But you should know who made the discovery and what the discovery was. Like, for example, you should know that Louis Pasteur invented pasteurization. You should know that Louis Pasteur came up with the concept of aseptic technique to keep your culture pure when you're growing it in a microbiological lab. You should know that Dr. Koch came up with colony streaking on a solid media, which was an easy way to get a pure culture. And then Dr. Koch also made Koch's postulates, the steps you take to show that a given organism causes a given disease. And along with that, Dr. Koch also showed the first bacterium that gave a disease. So he's the first one to prove the germ theory of disease, that germs can cause disease. And he showed that Bacillus anthracis can cause the disease anthrax. Okay, so you should know things like that. Any question about any of that? So I guess the importance is you should go over the history of microbiology. All right, if there's no questions, I'm going to end it here. Good luck on your final. If you have any questions, please email me. I'm here to help you. All right. Bye.